Kaylee Mortl. Hello, I have created a presentation examining the role of the pancreas and of the liver, two very important accessory organs of the digestive system, in regulating digestion with a focus on their coordination. First, it is essential to have a basic understanding of the functional anatomy of both the liver and pancreas before we dive into each of their roles in digestion and how they coordinate these roles. As you can see here on the right, we have a very nice diagram illustrating the structure of a liver lobule, a building block of liver tissue. Each hexagonal hepatic lobule, as you can see here, consists of a central vein, hepatocytes, or liver cells, and 6 portal triads, each containing a bile duct, hepatic artery, and portal vein, with a capillary network called hepatic sinusoids, which drain into the central vein. Interestingly, each human liver is estimated to contain roughly 1 million of these building blocks that allow for a blood supply to enter and leave the liver and for the drainage of bile. Now, on the left side of the slide, you can see a diagram illustrating the anatomy of bile ducts of the liver. As you can see, the liver has both a left and a right hepatic duct inside of it, both of which converge to form the common hepatic duct, leading to the common bile duct that not only receives bile from the gallbladder, but also converges with the pancreatic duct and ends inside of the duodenum of the small intestine. The pancreas is both an endocrine and an exocrine organ. It secretes hormones and assists with digestion. As you can see in this diagram, the endocrine portion of the pancreas consists of pancreatic islets that contain alpha and beta cells. The alpha cells secrete glucagon, a hormone that stimulates glucose production in the liver. The more numerous beta cells produce insulin, a hormone that allows the cells of the body to absorb glucose and promotes glucose storage in the liver when there is more present than is needed at one time. Moving on to the exocrine portion of the pancreas, which accounts for about 80% of the pancreas's mass, the cells that secrete digestive enzymes, which we will soon discuss, are called acinar cells, which are depicted in bright yellow on this diagram. The small green ducts that the cells surround in this diagram serve as the pathways by which secretions reach the central pancreatic duct and then empty into the duodenum via the common bile duct. Okay, now that we have an understanding of the functional anatomy of both the liver and of the pancreas, we can begin to discuss their roles in chemical digestion in the duodenum. First off, it is important to recognize that the duodenum itself secretes a number of hormones in response to the entry of chyme from the stomach via the pyloric sphincter. The hormone secretin stimulates the production and then the secretion of bicarbonate ions by the pancreas, which makes the pancreatic juice secreted into the duodenum have a basic pH of around 8.3. This basic pH is favorable for the activity of pancreatic enzymes. Next, we have the hormone cholecystokinin, or CCK, which stimulates production of pancreatic enzymes, stimulates the release of bile from the gallbladder, and relaxes the sphincter of Oddi, which allows pancreatic juice and bile into the duodenum from the common bile duct. Next, gastric inhibitory polypeptase, or GIP, promotes the release of insulin from the beta cells of the pancreas. And finally, vasoactive intestinal peptide, or VIP, is thought to regulate the composition of bile and pancreatic juice, as well as blood flow in the tissues of the gastrointestinal tract. Now, the major role that the liver plays in chemical digestion occurring in the duodenum is the release of a fluid called bile. Bile consists of water, bile salts, cholesterol, and phospholipids, as well as pigments like bilirubin that come from the breakdown of red blood cells, which occurs in the liver. Bile salts emulsify or organize fats in the duodenum by breaking larger fat droplets into much smaller fat droplets that can be broken down by pancreatic lipase for absorption. Moving on to the pancreas, as previously mentioned, the pancreas releases pancreatic juices that contain digestive enzymes. As a review, the bicarbonate ions in the pancreatic juice neutralize the acidic chyme with the basic pH of about 8.3. Now, each of the following pancreatic enzymes perform specific digestive functions. Pancreatic lipase breaks down triglycerides into fatty acids and glycerol for absorption, like salivary and gastric lipase. Pancreatic amylase breaks down carbohydrates, or starch, into disaccharides and trisaccharides, which are eventually converted into glucose for use in the cells of the body. This function is like salivary amylase. Pancreatic proteases, like trypsin, function to further break down proteins that have already been denatured in the stomach into amino acids. And lastly, pancreatic nucleases, DNase and RNase, are enzymes that break down both ingested DNA and ingested RNA into their nucleotides for recycling. This slide exhibits a diagram that I'm going to break down and explain in order to illustrate how the pancreas and liver work together to regulate blood glucose. Starting with the process of lowering high blood sugar, beta cells of the pancreas are stimulated to produce and release insulin into the bloodstream. Next, the presence of insulin in the bloodstream stimulates glucose uptake from the blood in the cells of body tissues, primarily muscle and adipose cells, thus lowering blood sugar. Additionally, insulin stimulates glycogen formation in the liver to lower blood sugar, as glycogen is the stored form of glucose. Alternatively, in the case of low blood sugar, alpha cells of the pancreas are stimulated to produce and release glucagon into the bloodstream, which stimulates glycogen breakdown to glucose in the liver, thus effectively raising blood sugar. These homeostatic mechanisms function in a healthy human to keep blood sugar within its homeostatic range. However, in a condition called diabetes mellitus, the body either cannot respond properly to insulin or cannot produce a substantial amount of the hormone. Now, there are two types of diabetes mellitus. Type 1 is characterized as an autoimmune disease in which the beta cells of the pancreas are destroyed and thus unable to produce any or a substantial amount of insulin. On the other hand, type 2 is characterized by insulin resistance by the cells of the liver, as well as muscle and adipose cells. In both cases, the patient must receive insulin injections to assist with the maintenance of blood glucose. This slide depicts a pathology that has negative effects on both the liver and the pancreas called sphincter of Oddi dysfunction, or SOD. In this condition, the sphincter of Oddi, the smooth muscle surrounding the end of the common bile duct and pancreatic duct, does not contract and relax normally. Usually, the sphincter relaxes during a meal to allow for the flow of bile and pancreatic juices into the duodenum. This condition can thus cause bile and pancreatic juice flow to become obstructed and can lead to pain, potential pancreatitis, and inadequate chemical digestion.